Hello, my name is Gema Torrijos, and I'll be presenting neuroprotective and symptomatic effects of cannabinol in an animal model of Parkinson's disease, written by Giuliano et al. Now for the research background, as you all may know, Parkinson's disease is characterized by progressive damage to the substantia nigra parts and factor. It's also widely known that glial cell activation contributes to the progression of neuron damage. Particularly, microglia and astrocytes have pro- and anti-inflammatory phenotypes that respond due to damage. Also, the researchers decided to use cannabinol, or CBD, due to its non-psychoactive, anti-inflammatory, and antioxidant effects. Now on to the research questions. The researchers had three primary questions. Firstly, does CBD actually help? Is there a reduction of degeneration within the nigrostriatal pathway when using CBD? And does motor function improve with the use of CBD? Secondly, they wanted to see the effects of CBD on glial cells. How does microglia and astrocyte cells change in response to CBD? Thirdly, they wanted to see how does CBD modulate astrocytes. Does CBD increase TRPV1 and glial expression in the substantia nigra? And does CBD increase CNTF and astrocytic cells of the substantia nigra? They did this using immunohistochemical staining. This is used to localize specific antigens of interest on tissue. Firstly, primary antibodies bind to the antigen. Biotin-tagged secondary antibodies bind to the primary antibody. This then allows stripped avidin peroxidase complex to bind to the biotin. Imaging analysis software can then be used to measure the puncta and compare. Now for the results of the first experiments, they found that cannabinol treatment attenuates nigrostriatal degeneration and improves motor performance. To do this, they used 12 rats per group and injected them with 6-OH-DA on the right side. This then caused dopaminergic loss of neurons on the right side of the brain. In the control group, you can see that 70% loss of dopaminergic in the striatal terminal and 70% loss of dopaminergic neurons in the SNC. This was contradicted by the CBD treatment group. In the CBD treatment group, they're given 10 milligrams per kilogram per day of CBD after 24 hours of surgery to 28 days. This then led to 55% loss of dopaminergic striatal terminal and 50% loss of dopaminergic neurons in the substantia nigra pars compacta. This is a 21% reduction of neurons. They also wanted to look at the effects of CBD on motor behavior. They did 3 tests. In the cylinder test, the rats were placed in the glass cylinder. The number of times the rat reared up to touch the cylinder wall was measured. To explain a little more, the rats were injected on the right side. The left side was considered the Parkinsonized side, and that's the contralateral side. The ipsilateral side, however, was the one that was showed to have preference and full use in both the control and CBD group. However, in the CBD group, they saw a 34% reduction of ipsilateral preference. In the apomorphine-induced rotational test, rats were given an apomorphine injection. This induces rotational motor response to the contralateral side of the lesion. They saw a 54% reduction with the CBD treatment group. Lastly, the rats were administered a rotorod test. The rats were pre-trained using a fixed speed and accelerating rotorod. The time on the rotorod was measured. Sadly, no improvements were seen in this experiment. Secondly, they saw cannabinol modulates neuroinflammatory processes in the SNC through preferential action of astrocytes. They first looked at astrocytes. In general, neurodegeneration is characterized by increased glial cell density. They used IHCs and compared CD11B plus to TH. CD11B is a microglia marker, and TH is a dopaminergic neuron marker. They saw no difference in the CD11 cells and cytotoxic M1 or cytoprotective M2 cells. So they went on to look at the microglia morphology. In this experiment, they saw an 8% increase in the number of endpoints found, but and a slight increase in the length of the branching. But this was not significant. In astrocytes, they used GFAP, an astrocytic marker, and saw an 8% reduction in the SNC in the CBD treatment group. Now, comparative to the control group, there was no difference in the phenotypes of the cells. Now on to the third experiment. Cannabinol induces the activation of TRPV1 and CNTF cascade in astrocytes. Now, why are we looking at TRPV1? TRPV1 is a CBD is a TRPV1 agonist. TRPV1 is characterized by the activation during anti-inflammatory response. In the CBD treatment, they saw an increased TRPV1 receptor expression in astrocytic cells, but no change in the microglia. This was then confirmed using a different technique called Western blot. In Western blots, they separate and identify proteins based on molecular weight and run it through a gel electrophoresis. The bands have been compared. In both experiments, there was a significant increase in the TRPV1 receptor expression, as you can see on the right side of both the Western blot and IHC graph. They also saw increased ciliary neurotrophic factor, abbreviated CNTF. CNTF promotes survival of neuronal cell types after injury. They saw an increased CNTF expression in astrocytic cells in both the IHC and Western blot, but no change in the microglia. As a take-home message, CBD-treated rodents showed significant reduction in striatal terminal degeneration and cell body loss in the SNC compared to the control group. This was accompanied by a recovery of motor performance. Chronic CBD treatment led to decrease of reactive astrocytes in the SNC, but there was no difference observed in the microglia. Speculatively, the reduction of active cells may consequently release pro-inflammatory factors and account for the therapeutic action of CBD. These pro-inflammatory factors might act on TRPV1 receptors. CBD-treated rodents showed an increased expression of TRPV1 only in astrocytes. Activation of TRPV1 receptors enhances endogenous neuroprotective response promoted in astrocytes through the production of CNTF. As Giuliano put it, CNTF released by astrocytes after CBD-induced TRPV1 activation might promote the survival of dopaminergic neurons, resulting in recovery of motor performance. Thank you.